The overall goal of this procedure is to detect and track reversal of the cell death process after caspase activation in live animals, using Drosophila melanogaster as a model. In response to death stimulus, cells undergo programmed cell death such as apoptosis, which has traditionally been considered as an irreversible cascade leading to cell death. Interestingly, however, after removal of the death stimulus, some dying cells can reverse the cell death process even at the late stage through a newly discovered cell recovery phenomenon called anastasis. Live cell microscopy shows that apoptotic dying cells display unique morphological hallmarks such as plasmid membrane blebbing, nuclear condensation, and cell shrinkage. In general, these cells are expected to die. However, after washing and incubating the dying cells with fresh medium, they undergo anastasis, recover, and can later divide. It is difficult to detect and track anastasis in live animals because the recovered cells can be morphologically indistinguishable from the surrounding healthy cells. To solve this problem, the mammalian CaspaseTracker Biosensor system was developed to identify and track the cells that reverse the cell death process after execution or caspase activation, the hallmark of apoptosis. This biosensor system consists of two components, the caspase activatable transactivator RTTA and the CreLox-P-based RTTA activity reporter. In healthy cells,